Jonathan Milner probably needs very little introduction to many of you. I was going to say he's the poster boy, but that doesn't sound quite right, does it? Sorry, Jonathan. Jonathan is an example of the Cambridge phenomenon, starting in the lab, starting his first company, which has been hugely successful, turning from a scientist. You don't ever stop being a scientist, but turning into a businessman, and now being very active in the Cambridge ecosystem, helping other companies to start and get rolling. So we're absolutely delighted to have him with us tonight, and now I'm going to get off and let him get on.
So when I was invited to talk by Andrew, he said, what are you going to talk about? And the first thing that came into my mind was building businesses, because that's what I've been doing. But as Mike quite rightly says, I'm a scientist, and once you're a scientist, you're always a scientist. And then Andrew said to me, well, Jonathan, you're a nerd, and your audience is nerds, so we're all nerds here. So I thought, right, if we're all nerds, I'm going to talk about science and research and what this society is all about, which is the application of research. So I'm actually not going to talk about, so I'm just going to get the apologies over to begin with, because I'm actually not going to talk about building world-class life sciences businesses. I'll touch on it, but that's all I'm going to do. So apologies, first of all, for that. And what I will talk about, and I'm going to talk about the golden age of biology a little bit, not very much, but I'll just touch on that. I'm going to talk about some of the areas that I'm involved with that are really exciting, well, I believe are exciting, and that I've been following for a number of years now. And this is what I want to talk about for the bulk of my talk. I'm actually going to be talking about diseases of aging. And I've been fascinated by this for many, many years. And I think now is the time when we can bring some of this research together. And I want to share that with the audience tonight of where we are with tackling diseases of aging. And also in new approaches in aging research. And then, because we're all nerds, and we're engineers as well, and biology ultimately is just an engineering problem. I thought I'd talk about mitochondria. Mitochondria, for me, are one of the most fascinating things in biology and one of the most exciting research areas. And again, I thought that the audience would be interested in hearing about that. And we're going to go into something about how mitochondria function, which personally I find fascinating, and I hope you do too. And then I'm going to introduce a term that I think its star is rising, and this is something called mitochondrial heteroplasmy, which lies, it seems, at the core of so many diseases of aging, and possibly even aging itself. So that's what I'm going to talk about. And with aging, you need some good news. There's nothing you can do about aging, so you need to have some good news. So we'll start with some, well, actually, before I get into aging, I will just very quickly talk to you about what I've been doing since, just to sort of recap, I founded this company called Abcam. I was working as an academic, working on breast cancer research at the Gurdon Institute. And then after 16 years of being CEO of Abcam, I stepped down. I'm still on the board in September 2014. And one of my big aims was to actually spend less time with my family. So I launched myself into a load of exciting areas that I thought was exciting. And being in this golden age of biology, I was actually just overwhelmed with opportunity. And so I've chosen 5 areas that I'm really actively involved with and started companies in. And I'll just very briefly share these with you. So the first one is immunotherapy. And I've got some colleagues at Addenbrooke's, oncologists, and I can honestly say that I've never seen them so excited about this area of medical breakthrough. It's incredible what's going on here, and there's actually people being cured using these immunotherapy treatments. So just to remind you, what these immunotherapy treatments do is they harness the power of our own immune system to attack cancers. So it unleashes our immune system to attack cancers. And the cure rates are modest at the moment, 10 to 20%, but they're getting better and better and better as we explore and discover how to do these treatments even better. So this is hugely exciting, but I'm not going to talk about that today, but just to say that some of the companies that I'm involved with that are leading the way here, Abcam's got a fantastic anti-PD-L1 antibody, if you're interested in that, and Horizon and a new company that I started with Chris Torrance, who started Horizon, called Foremost. The next area we've heard an awful lot about is genomics research. And being a nerd, as soon as I could afford it, I got my genome sequence, and I'm sure many of you in the audience have had your genome sequence. If we look back, not very long ago, it was billions. The first genome to be sequenced cost billions, and now it's under $1,000. And this is really revolutionizing healthcare. And it's not just genomics, it's also proteomics, transcriptomics. There's a lot of omics data out there, and making sense of that omics data is quite difficult. And so I got involved. I launched myself into getting involved and helping to start up with a young scientist called Fiona Nielsen, a company called Repositive. And Repositive, to sort of summarize it, it's like we want to be the Google of genomics data. So it's the place for scientists to go and search through all the vast amounts of genomics and other omics data that's out there. I'm not going to talk about that anymore today. And then the next area that I'm involved in is stem cells. And what I love about biology is that when I first started my career back in the early 80s, there was a dogma out there, and that dogma was that our cells, once they differentiate, so once you've got your fertilized embryo, and those cells go on and differentiate and become your heart cells, your liver cells, your skin cells, your muscle cells, your brain cells, your eye cells, etc., there's no going back. And then a Japanese chap called Yamanaka discovered there was a way of actually turning our differentiated cells back into pluripotent stem cells. And this is absolutely revolutionizing biology and medicine. And it's not just in regenerative medicine, it's also in drug discovery. And the exciting thing is that pharmaceutical companies are now using these, what we call patients in a test tube, to actually test their drugs on. So you don't have to go into animals or anything like that. So you can vastly reduce the attrition rate of drugs coming onto the market by pre-testing them in these cells that are grown as like patients in a test tube. And I have a number of companies there. Horizon again, and Axel and Definigen. But I'm not going to talk about stem cells. And then I was very fortunate to meet a young entrepreneur scientist called Tim Guillaumes. And he said, Jonathan, I've got this really interesting way of being able to repurpose drugs for rare disease. I said, oh yeah, tell me about it. And he did, and I got fascinated by it. And it's using transcriptomics. It's called transcriptomics matching. But it uses basically machine learning, artificial intelligence, to repurpose existing drugs to treat rare disease. And Tim's big vision here is to he calls it n equals 1. So in statistics, the number, the cohort number is n, and he wants to be able to say, for somebody with, you've got this person with one person with the rarest disease in the world because it only affects one person. But we can find the drug to treat that one person. So we calls it N equals 1. So I'm having enormous fun with Tim on that one, but I'm not going to talk about that today. So that's HealX. And then finally, I've been, as I said, really fascinated by diseases of aging and mitochondrial research in particular. And this is actually, so I've got a couple of companies there. Foremost, again, has got an aging program. It's called Forever. This is a company which I'm going to tell you about a bit later on in the talk, which is I think, at the leading edge of mitochondrial research and this thing called mitochondrial heteroplasmy. So that's what I'm going to talk about. I'm going to spend most of my talk on that. And this is where I want to sort of start off on a positive note, because we don't want to get ourselves depressed. So let's look at some statistics. Well, more and more people are living longer and longer. So the number of people over the age of 90 has virtually doubled since 1985. And that's all good news. And the other great news is that we're living healthier. So the number of people in age 65 plus that are actually in active employment has virtually doubled in the last 20 years. And this trend will continue. This is fantastic news. And then this graph is wonderful. So this is even better. I mean, this just gets better and better and better. And this graph is going to take a little bit of explaining. So we've got over here the percentage of the population alive by the date that those people were born in. So we'll start off with historic data for somebody that was born in 1851. What could they statistically expect to live to? Here's my clicker's just gone. Anyway. Right, so, and that's the curve we get. So that's historic data. So by the age of 10, you've got a 30% chance, basically, of not being alive. So it's pretty poor, really. And then not many people are living past the age of 90, and it's pretty steep, this curve. Things start to get better, however. So in 1871, things are getting much better, and there's hygiene and germ theory and all of these things coming into play. And 1891, it's getting even better, and then, here, this is astonishing. I mean, today, we couldn't accept this. But if we go back only 200 years, 40% of births ended in death before the age of 1. That's totally unacceptable today. It's a tragedy when one baby dies, but then it was 40%. So it was incredibly high infant mortality. And then we're getting into the realms of people that are still living. So these are the people that were born in 1911. And then lifespans are increasing. And then it gets even better. So people are less likely to die, much less likely to die in younger age. And here you've got people that were born in 1931, and half the population, which is about right, are living to, what's our average lifespan? Something between 80 and 85 years now. So this is absolutely wonderful news. But it gets better, because if you're born in 1951, you can expect to live even longer. And again, in 1971, even longer. To the point where, actually, the people born then, in 1971, and in fact, probably 1951 as well, half the people born then are going to be alive at the age of 100. So let's extrapolate this forward a bit more. So these are people born in 1991, and then we get to 2011, and it keeps getting better and better. Now, what happens if you extrapolate this even more and you think, well, science is going to come up with all sorts of treatments for diseases of aging and everything. And this is not beyond the realms of possibility, that we see people living longer and longer and longer with really, really very healthy lives. And then we're all sort of dropping dead at the age of 140. And the wonderful thing about this is, if it was to come to pass, it would also solve one of the biggest problems that we face today, which is healthcare costs. The burden of healthcare costs is huge, as we know. And 80% of healthcare costs actually fall within the last sort of 20 years of life. So you get what people are calling the lifespan dividend. Actually, it's not really a lifespan dividend. It's more of a healthspan dividend, as we shall see here. So this is where we're currently at. And unfortunately, as we're going to come on and see, and this is part of the problem, and this is where it gets a bit more depressing, after the age of 50, we start to get diseases of aging. So this is this red color here, and the prevalence of them here. And I remember Tony Kouzaridis, I was complaining once in the lab. This is before I started Abcam. And I was complaining to him about my research wasn't going well. And he turned around to me and he said, Jonathan, you know what? Life is shit and then you die. And I thought about this and I thought, well, actually, really, life isn't that shit. If you're healthy, it could be pretty good. So, and it was good though, because it made me stop complaining. But, you know, life can be pretty shit in the last years with these diseases of aging. So what if you can actually not have this extended period of morbidity, but compress this so that, you know, life is shit and then only the last 2 days, 2 years, or even 2 days are even better. But very, very short amount of time at the end of your life, and then you die. So this is the sort of thing that interests me. And it was always a bit of a sort of pipe dream, I suppose. But actually, some of the research now is showing that we can do this. One of the problems has been, and I put this slide up here, because we need to think of these diseases not in isolation, but actually diseases of aging. And this is a really important point, because we've really missed a trick here. And I think governments, pharmaceutical companies have missed a trick by thinking of these things in isolation without thinking, well, what is the biggest risk factor for these diseases? And all of these diseases are all diseases of aging. And if you don't believe me, I've got the data, because we're all nerds and we love data. So I don't expect you just to sit there and go, yeah, all right, believe me. But here's some data. So this data is from the Alzheimer Europe, 2009. So a reputable source. And if we look at cardiovascular disease rates, you can see that about the age 50, just like I showed on the other slide, cardiovascular disease starts to really kick in. And it's kicking in, so the incidence is going right up to the point where half of us men, we've got some form of heart disease by the time, well, nearly half of us, by the time we're 75 plus. It's slightly better news for women. People don't really know why. I expect some people in the audience have got some theories, but I certainly don't know why. And again, it's the same for cancer. And we seem to get to this point of around about 50, and then cancer rates accelerate and really kick off. And in fact, you know, sadly, my father was, when he died of cancer, he got lung cancer, he contracted that at the age of 52, and he died at 54. And again, the prevalence of it increases faster for men than it does for women. Okay. So if that doesn't convince you that these diseases are not diseases of aging, then I don't think anything will. And then the final graph I want to show, I think, is very interesting. And this is the rates of dementia. So this is the prevalence here, and this is by age. And you can see that dementia actually starts to rise later than these other diseases, and the cardiovascular and the cancer diseases. So if you can survive these, sadly, you know, many of us are in for a future of dementia here. And this is especially interesting to me because I don't know how many of you in the audience have done 23andMe. So you can do a genetic test, and it will tell you what your APO E4, E3 status is. And if you carry the E4 gene, like myself, I'm at a much higher risk of developing Alzheimer's. So my lifetime risk is something like 29% of developing Alzheimer's, as opposed to the general population where it's about 9%. But you can see here something very interesting going on, which is that females are getting dementia at a higher rate, prevalence, than males. And nobody knows why that is, but I have a theory. My theory is that all us men are being killed off by the cardiovascular and the cancer, and the cohort that is actually surviving the cardiovascular and the cancer has a healthy set of genes, which is also enabling that cohort there to be less prone to dementia. That's just my pet theory. There's absolutely no evidence for that whatsoever, but anyway. So that's all a bit depressing. And this is another depressing slide. We've got heart disease here, still the biggest, so the two biggest causes of death. We've got the heart disease and the cancer here. So the reason I wanted to show this is because we do get obsessed with curing cancer. We do get obsessed with curing cardiovascular disease, and we've had some incredible successes with both of those areas. But there still remains the aging problem behind it. So an interesting fact for you, if you could wave a magic wand and just suddenly cure all cancers, what would happen to the average life expectancy of the human race? And the first time somebody asked me this, I said, it would be 20, 30, 40 years. The answer is actually 3 years. So on average, we would all gain another 3 years if cancer was cured. Now, that doesn't mean we shouldn't spend an awful lot of time, as I did in the lab, trying to cure cancer and coming up with therapies for cancer, because the cohort of people that do get cancer, if they get cured, they'll get an extra approximately 14 years of life. So that's definitely worth doing. But on an average, for the people who don't get cancer and everybody, it's only going to extend life 3 years, because something else is going to get us. I've put this very complicated slide up, but I'm not going to go into it. All I want to say here is that something that we already know, that the drug industry is in trouble because it's costing much more, like 50 times as much per drug that it costs now than back in the 1950s. And the number of new drugs coming onto the market is fewer and fewer. And one of the reasons for that is because drugs companies are looking at trying to cure these intractable diseases of aging one at a time without actually looking at aging itself. So you'd have heard recently the news, I think it was last week, it was Pfizer pulled out completely of research into Alzheimer's, or developing drugs for Alzheimer's. And all those drugs that are put into Alzheimer's, they have all failed completely. Sorry, this is another graph. It's just showing that only less than 1 in 10 drugs that go into clinical trials actually make it onto the market. So a huge attrition rate. So back to Alzheimer's. This is quite remarkable and makes me happy. Although what I'm about to tell you, you could probably guess for yourself, because I think it's what we all really know. But the one big breakthrough in Alzheimer's has come from this guy here. And he's a hero of mine, Dale Bredesen. And basically what he said was, do you know what? I'm just going to throw everything I know about Alzheimer's at my Alzheimer's patients and see if I can reverse their Alzheimer's. And everybody laughed at him. They said, there's no way you can reverse Alzheimer's. Once Alzheimer's set in, you cannot reverse it. He said, no, I don't believe it. All I'm going to do, I'm going to treat them with what I call the Bredesen Protocol. So he did the Bredesen Protocol, and the Bredesen Protocol is this. Here it is. It's a healthy, low GI diet. Not too much food. Not much carbohydrates. Keep your insulin levels low. It's more sleep. It's lower stress. It's more exercise. And he looked through all the literature and came up with 23 supplements that are available, that are out there. Things like lion's mane mushroom, bacopa, vitamin D, turmeric, etc. And he put them all in a cocktail and he said, I'm going to give those to my patients. And the results have been quite remarkable. So all of these years of the drug companies trying to develop drugs and cure Alzheimer's have been completely blown away by this guy, just doing really simple stuff. And he's got a 90% success rate in reversing Alzheimer's. He's put 110 patients through so far. And if you don't believe me, because I didn't believe it, I thought it was too good to be true, you can go and look at it. He's published it in Aging, June 2016, Reversal of Cognitive Decline in Alzheimer's Disease. So coming back to aging research, one of the problems is that aging is not actually recognized as a disease. And you could probably argue, well, probably it shouldn't be recognized as a disease. But then if it's not recognized as a disease, it doesn't get reimbursement by the FDA. And people are not putting enough money into it. So here's some figures for you again. 90% of US deaths, at least 80% of US medical costs, are caused by aging. And yet, if you look at the budget, which is 30 billion, how much of that is actually spent on aging? Well, they do have a National Institute of Aging budget, but most of that money is actually spent on, again, single diseases, such as trying to tackle Alzheimer's or cancer. So the amounts actually spent on aging biology is only 150 million, and actually on translational research, where they're putting things into clinical trial, is minuscule. And then I asked permission of a chap called Aubrey de Grey that many of you have heard of. Many of you probably will know him. He's a Cambridge guy who's obsessed with curing aging. And his budget for the SENS Research Foundation budget is only 5 million. So these amounts of money are tiny for tackling the size of the problem. And that really does need to change. But what's going on, even with this small amount of money, what's actually going on? And what I want to introduce you to here is 3 figures in aging research who I think are really, really important. And you may have heard of them, you may not have. The first one is the chap I just mentioned, and here he is, Cambridge's very own Aubrey de Grey, although he is in California now. And he has this research institute which he started, which is Strategic Engineering for Negligent Senescence Research Institute, and it's all about aging. And he has some very interesting, controversial ideas, and 10 years ago, everybody thought he was a bit, not quite right in the head. Trying to look for a polite word here. But anyway, it turns out that he is right, actually. And much of what he says is absolutely right. And the new approaches to aging research, he's absolutely spot on. And when he talks now, people actually do listen to him. And he said it's quite possible that the first person to live to 1,000 years old has already been born. I have no idea if that's true or not, but he sees aging as an engineering problem. And we're all there's a lot of engineers in the audience, so you can sympathize with this. And his analogy is, well, you can keep a car running forever as long as you maintain it well. There's no reason why it can't go on forever. And the part of it, we're machines, and engineering will solve the problem. The next chap is really interesting. He is an Australian called Professor David Sinclair, and you may have heard of him through the red wine and resveratrol story. But there's so much more to David Sinclair than just that story. So he splits his time between the University of New South Wales in Australia and Harvard Medical School. And he started a company called Certris, sold that to GSK for $700 million, and GSK haven't really done anything with that, sadly. But he's gone on to be a real sort of proponent for aging research and one of the leading figures there. And the next guy is, you probably haven't heard of, he's a bit of a hero of mine, Dr. Matt Kaberlein. And actually, he was in the same lab at Harvard as they were contemporaries together. So him and David Sinclair were contemporaries together in the same lab. And he discovered another pathway. So David Sinclair was one of those discovered the sirtuin pathway. And Matt was one of those that discovered what's called the mTOR pathway, as we're about to see. But before we do that, we have to sort of think about a definition of aging. And I unashamedly borrow this from Aubrey de Grey. I did actually ask him permission, because this is his aging. And it's what happens to the body as we just go through everyday life. And we're sitting here metabolizing now, and we're creating damage to ourselves. And it's just a normal part of the body's normal operation, is that we're metabolizing, and this is creating free radical species, which are causing damage to the body. And of course, we can tolerate quite a bit of damage, actually, if you think about it. I mean, we hit our metabolic peak of fitness sort of probably at the age of 20, 25. But then we can go on for years and years and years, perfectly fine, with plenty of damage. But at some point, the damage causes too much, and it causes, and we get to disease and disability. So you can think of it in the following way, and that is, here we have metabolism, and as an inevitable result of running the car, if you like, you're going to damage, or running the cells, you're going to generate damage. And then too much of that damage, and it leads to diseases of aging, such as your cancers, your Alzheimer's, your diabetes, etc. And the approach that we've been taking in the past is one, first of all, through can we try and limit the damage being done through things like gerontology. And if you remember, probably about 20, 30 years ago, there was huge enthusiasm for antioxidants. And there were antioxidant trials of vitamin E and vitamin C. And then those trials were very quickly abandoned because it became apparent that the placebo group were living much longer than the people that were actually getting the vitamin C and vitamin E. So this idea of sort of slowing down the hasn't really got us anywhere at all. And again, this isn't really getting anywhere as well. Trying to treat once the damage is done, trying to prevent that damage from actually going into causing pathology, going into causing the disease. So treating the individual diseases themselves without thinking about treating the whole of aging. So this is a bit of a problem, really. But as Aubrey de Grey says, and many people are now coming round to his way of thinking, what if you could do more maintenance and repair? You can repair the damage and maintain it, and in theory, go on and on and on and on and on. So this is now seen as the most promising area in aging research. So finally, people are taking Aubrey, David Sinclair, Matt Kaberlein actually seriously, and coming and seeing that this is the way forward. So we want a new approach to tackle the problem of aging, prevent or delay the onset of aging diseases. First of all, the FDA, the Federal Drugs Administration in the US, because they are the guys who actually lead the way, and then the UK follow. But so they really need to do this first of all. They must recognize aging as a disease. And the really good news is that if you're recognizing it as a disease, which they weren't doing, then you need to find drug targets. And one of the biggest criticisms of saying, well, aging is a disease is there's no drug targets. That's actually not true. There are drug targets. And there's one that was discovered in 1997, which seems to be a master regulator of the cell in all the processes of the cell that go wrong in aging. And it's a gene called Clotho. And that was named after one of the three Greek goddesses of the fates that wove the thread of life. And here she is, weaving the thread of life. So what does Clotho do? And when you look at Clotho functions, if you upregulate Clotho, you have a positive impact on all of these things, which, funnily enough, are all involved with aging. So the regulation of ions within the cells, that's one of the things that goes wrong in aging. Insulin metabolism goes to pieces. That's why diabetes is a disease of aging, and people get fatter as we get fatter as we get older. Glucose metabolism, again, related to insulin. It's got antioxidant effects, laying down of fat, and generation of new blood vessels. So it really is a very exciting target. So there's no excuse anymore. There are targets coming out that can be used to develop drugs to. No drug companies are actually developing drugs to these, though, because they're not going to get reimbursed for any drugs that come to market. And we've seen the problem of how expensive it is for drugs to be developed. But there are a few startups working on Clotho. So if aging is an engineering problem, can we boil it down to a few processes within cells that are going wrong, and then gradually sort of work our way around and sort of cure each one of these processes and then fix aging for good? And this came out in 2013, and most people agree now this is a really good basis for thinking about the problem of tackling aging. So where would you sort of look? Well, in aging, you get cellular senescence. And as we all know, as we age, we have fewer cells because they're not being replaced. They're dying off. And so people actually, you can see it here, people are actually visibly shrinking. They may get fatter, but the number of cells mean they're visibly shrinking. And you get the wrinkles and everything associated with aging. Stem cells, which are meant to be replacing the cells that are dying off in senescence, themselves get exhausted. Cells are not communicating with each other, and you get dysregulation there. You get genomic instability, telomere attrition, which is, again, related to cellular senescence. You now get a really well-defined set of epigenetic alterations. If you remember, just to remind you what epigenetics is, it's where you've got your genetics is your hardwired code that's in your DNA, it's in your chromosomes, and on top of the chromosomes you've got all the factors, the histones, the things that wrap the chromosomes and go around the genes, and these get epigenetic marks on them, and they're caused by the environment. So you accumulate epigenetic marks on the epigenome as you go through life. So you can there's actually a very exciting new piece of work which shows there's what's called an epigenetic clock, and you can look at the epigenetic changes in the epigenome here. Loss of proteostasis, so proteins at the heart of cells, as you know, and if they are not properly folded, again, the cells get into real trouble. And in fact, loss of proteostasis is the partner, really, for mitochondrial dysfunction. And these two takes two to tango, if you like. So you get loss of protein function, and that affects the mitochondria. The mitochondria aren't as healthy. Or you've got unhealthy mitochondria, and you get loss of proteostasis. And then, of course, there's deregulated nutrient sensing, where the cells just have no idea what's going on in terms of their glucose metabolism. And of course, mitochondria lie at the heart of all of that. And we're going to come on, and I think probably where we should be starting is here, because mitochondria really have, they really do cross all of these different characteristics, as we shall see. So one of the most incredible discoveries, which really went under the radar, was that this was actually discovered many, many years ago, but people didn't really believe it. And it was people, because all these scientists are working with little white mice. And sometimes scientists, I don't know why, decided not to feed them. And they found that the ones that they weren't feeding just seemed to live longer. And then slowly, sort of it dawned on people, this is incredible. So we're not feeding these mice as much, and yet they're living longer. That is really bizarre. And consistently, this is one of the only environmental things, aside from some drugs, as we're going to visit in a minute, that consistently actually extends lifespan. And not just in mice, in nematodes, in Drosophila, even in monkeys. Nobody knows if it works in humans or not yet, although there is a Society for Caloric Restriction, which if you want to join it, you can, but I would find that utterly miserable, if I'm honest with you. Anyway, here's the data, and it's quite remarkable. So these are the survival. So your mice are living, on average, just over sort of 35 months. That's the average lifespan. Well, it's actually less than that, isn't it? It's just less than 30 months there, 50% survival. And you're getting this enormous increase in lifespan when you're giving them, the poor little things, only when you're reducing by 65% their food. So you've got a 65% caloric restriction here. Although the ones on 55% seem to be doing just as well in terms of longevity. So this is astounding piece of work, and it's been repeated again and again and again in many species. And if you're interested in the Caloric Restriction Society for humans who practice caloric restriction, here is the website here. So this observation has been at the heart of what we now know about aging. And the reason for that is it led people like, it's not just David Sinclair, but people like David Sinclair and Matt Kaberlane to discover pathways involved with longevity. And these two pathways are the mTOR pathway and the SIRT1 pathway. And caloric restriction, it seems, is acting to reduce insulin, and it's acting to inhibit mTOR that way. It's acting on AMPK and mTOR that way. And then here, we'll come on and we'll have a look at NAD plus concentration. So NAD plus is a cofactor which transfers electrons in oxidative phosphorylation in the mitochondria. And if you don't have enough of that, you downregulate SIRT1. And this, so the effect of caloric restriction on these two pathways means that, and that's how they worked out why the mice were living longer. So it's not just about caloric restriction, because this is a wonderful piece of work. And it was done by an American who was obsessed with people who are living to 100 and above. And he wanted to find a control group of people with exactly the same lifestyle, but look at at what point the onset of disease happened. So these are matched controls. So these centenarians here, in this group here, are actually having a lifestyle which was matched for the controls, and that lifestyle was everything that you shouldn't do. Smoking, eating too much, some were obese, etc. So these lucky people here have got a good set of genes, which means that they're living longer, but also, as you can see along here, they're living healthier. So it's much, they're delaying the onset of diseases of aging quite considerably from the control group. So they're actually living longer and living healthier. And they form a really important part of aging research as well. So there's quite a few genes that have now been discovered that are associated with these centenarians, which are what are called longevity genes. And yes, you've guessed it, they all seem to go back and point back to the pathways of mTOR, SIRT1, caloric restriction. So everything seems to be pointing back to this. Now, a lot of the research has been done on mice, and this is fine, but we're not mice. In fact, we're not hyper-intelligent, pan-dimensional beings. We're humans. So can we actually trust this research from the mouse? I think probably yes, because the mitochondria are highly conserved across all the mammalian species. And these pathways are highly conserved. So actually, unlike sort of drug discovery, mice and other mammals are actually a really good proxy for what's going on in humans. So if we have a look at some of the things that have been going on, and bear in mind that all this has been done on a shoestring budget. It really has. And we can look at compounds that are being used to combat aging. And I've listed as many as I could. Well, these are the main ones here. So we've got Metformin, which is already being used. We've got a substance called nicotinamide mononucleotide, which is a supplement. You can buy this. We've got our friend resveratrol. That's David Sinclair again. We've got a homologue of glucose, 2-deoxyglucose it's called, and we've got a substance called rapamycin down here, which is already being prescribed as is being used as an immunosuppressant. So here we have some mouse data, and we have the effect of these compounds on the Healthspan. So are the mice healthier? We have the mean lifespan. So on average, how much are they living longer? And then we have what is the maximum lifespan. So if you've got some mice that are living much, much, much longer. And if we look at metformin, it seems that it has a really good impact on Healthspan. And in fact, this was picked up, first of all, because there was a study done on diabetics who were taking metformin, and it was noticed that they were suffering less cardiovascular disease and less cancer, and were actually healthier than the non-diabetic control group. And that's how the interest in metformin got going. What about this one? This is pretty good. This is got a good Healthspan, mean lifespan, maximum lifespan. It doesn't look so good, but still. Resveratrol, again, Healthspan, lifespan, didn't really do much. Maximum lifespan didn't do much. 2DG, Healthspan, great. Didn't really do anything for mean lifespan or maximum lifespan. Finally, we come to rapamycin, and that has an effect on Healthspan, lifespan, and maximum mean lifespan, and maximum lifespan. But they're all promising compounds. So what's going on? You'd think with all of that data there, there'll be so much work going on in that. Well, actually, there isn't really a lot. So in terms of human trials, there is one which is called TAME, which we'll come on to in a minute. With NMN, there's one going on in Japan, small study going on in Japan with the University of Washington. This is a really sorry tale. So Paul, well, actually not Paul, because he did sell his company for $700 million to GSK. But David Sinclair's Certris, the Certris program in GSK was cancelled after a number of years. And I think that was citing that they couldn't get a handle on the target for the resveratrol molecule and the resveratrol-like molecules. So that's been discontinued, sadly. This is still experimental. And this one's interesting because there's a project going on in dogs. And if you want to get hold of these things, well, metformin is prescription only. You can actually buy these, although this one is horrendously expensive at the moment. That's really cheap. Resveratrol is very cheap. You can't get that because it's experimental. And that actually costs more per gram than gold. So very, very expensive. And rapamycin is only on prescription, so you can't really get that one. And these are just the aging targets and mechanism, which I won't go into, but if you just quickly skim that, you can see it's all pointing back to caloric restriction, mitochondria, and the two pathways, the mTOR and the SIRT1 pathway. So these are very promising compounds, and we're going to wait and see what's going to happen there with the trials. This is the data from rapamycin. You can see that that's extending the lifespan of these. These are female mice here. So that's really good news. And here's a dog. And I'm a big fan of dogs. I've got 4 dogs. And they're not just man's best friend. They're also going to help us to live longer because they get many of the diseases of aging that we do, only it's vastly accelerated. So one of the problems of doing any trials in humans is, of course, we live a long, long time. So getting the results from those trials is going to take 20, 30, 40 years before we see the results of trials, and you just can't wait around that long. And here's my friend that we met before. This is Matt Kaberlane, and he's doing the dog aging project. And if you want to go to the website, you can, and you can sign up your dog. Although I tried that, and I think it's only open to US dogs at the moment. I'm still waiting to hear from them. And here he is with his dogs. And he's using the compound that I showed you that had the most promising mouse data, rapamycin, and he's using that at a low dose so that it doesn't suppress the immune system in dogs, and seeing what the effect on health and lifespan is in those animals. And here's a very a bit of a busy slide, so I apologize for that. I'm not going to read all these out, but this is a study done by David Sinclair and others. It's not just him. It's mean to say that it's just him. There's others that are involved with the study. But he used the precursor of the cofactor NAD+, and fed that to mice here, the NMN study. And on all of these factors, which can be a proxy for aging, the mice here were much better off, which is the green, than the control mice, which were fatter, aging more poorly, etc. So this is the study that's going on with TAME. So this is targeting aging with metformin study. Okay. So this is actually going on in humans. It's being done by the American Federation for Aging Research, and it's a charity. And this is the website here, www.afar.org, and it asks for donations for this trial to occur. And this is just what they're doing. They're doing, including 65 to 80-year-old people, they're trying to get to 3,000 people. They're going to do a double-blind placebo-controlled trial with the primary outcome of time to major disease. So, anecdotal data. Here is David Sinclair. So you think David Sinclair, being one of the pioneers of aging research, would be taking these drugs himself. And yes, he is. So he's actually taking a combination of metformin and NMN, and he went to this website called www.inside Tracker, and you put in a lot of your blood tests, etc., and it will approximate your biological age. So this is David's chronological age here, and it's going up over 3 months here. And if you put in all your data about your insulin levels, your glucose, your HbA1c, your cholesterol, it extrapolates it all in a big algorithm, and it comes out and it says you are this biological age, which is, of course, different from your chronological age. So David was shocked because he comes out at an age of 57 years old. But after 3 months of taking metformin and NMN, he was down to a biological age of 31. And anecdotally, he says he's never felt better in his entire life, and he's leaping around, and he's got his father on these drugs as well. Whether you believe it or not is up to you, but certainly a study of just n equals 1 is not going to do it for me. But good for him. It looks very promising, let's say. So I want to come on to the last part of the talk and just spend a few minutes looking at mitochondria. And as you all know, mitochondria are what we can consider the batteries within the cell. So when the batteries wear out, we're in real trouble. And here's a mitochondria, and you've probably seen many pictures of these. And if you remember, they're formed by the symbiosis of an early bacterium right in the dawn of life with a precursor eukaryote cell. And they get together, and that's how all of multicellular life sprang out. That's why we're not just bacteria on this planet. It was that symbiotic event of these bacteria inside our cells. And they have their own little piece of DNA in there. Most of the DNA that was in the early, when the symbiosis formed, migrated into the nucleus, but they still have remnants of DNA within them. And here's an Abcam antibody, which is staining the mitochondria. And actually, it's a myth, also about bioelectron microscopy, actually. But most of the time, mitochondria form this reticulate structure, this sort of network. And it's really only when they bud off is when you get these sort of little bacteria-like structures. So that's all lovely. And this is I'm not going to go into any detail here, don't worry. But all I wanted to say here, this is just such a fantastic achievement of humankind, actually, in being able to work out exactly how mitochondria function and how they generate the energy that we all need to live. And it's through this proton pump. So it's just like a battery. So what's happening is that these protons are being pumped out, and so you get a higher concentration of protons out here, and they zoom back across the membrane of the inner membrane of the mitochondria. And in that process, there's this incredible ATPase that spins round and round and round and creates ATP from ADP and inorganic phosphate. And it's that ATP molecule which has got the energy that's used for all of cellular processes. This is what powers us. And I just wanted to mention, again, coming back to Cambridge, another hero of mine. This is Sir John Walker here, and here he is in his lab. And he was one of the ones responsible for working out part of this pathway. And for that, he got the Nobel Prize for Chemistry in 1997. And this is what he discovered. And this is really why I love biology, and really why many of you will like this, because there's a bit of engineering in here. So at the heart of mitochondria lies this ATPase. I mentioned to you the proton pump, where you've got more protons out here. And so there's a proton gradient. And so John worked out the structure of and others as well, but John as well, worked out that the full structure of this. And as protons go through here, it causes this to spin round and round and round and round at about 130 revolutions per second. And in the process, it catalyzes this reaction to generate ATP. So at that point, it all looks a bit sort of molecular and techy, So I thought what we could do is just have a brief interlude of this beautiful sort of dive into mitochondria, where you can see a molecular animation of what it would be like. Because, of course, you can't see, even with the most powerful electron microscopes, this process going on. It's just a bit of a blur. But if we could really get down in there, what would it actually look like? And this is the best way, I think, of visualizing it. So this will take you through the mitochondria from the macrostructure. Then it will dive into the membranes. And then look out for the beautiful the proton pump and the ATPase and the revolution of the ATPase. And the protons are like little gold dots. And the ATP is, again, a gold molecule that's glowing as it's receiving its energy. So I'm going to shut up there, and we're going to enjoy this amazing video. Good. So you can see why biology, even at that level, can be hugely exciting. I hope you enjoyed that. So now I want to just end my talk in the next few minutes talking about what's going on with mitochondrial DNA and, excuse me, mitochondrial heteroplasmy. So when we look at the genome for the mitochondria, or the plasmid, as they call it, it's tiny compared to it's only 16,000 base pairs compared to the 3 billion base pairs that sits in the nucleus of our cells. And it's been well mapped out, although it's only been recently that the techniques for actually isolating the mitochondrial DNA away from the 3 billion base pairs of nuclear DNA has been, with next generation sequencing, it's been really possible to do this. And the thing that's been discovered is that many quite rare diseases, and some not so rare, such as Parkinson's, have mutations which map straight onto this plasmid. And so this plasmid contains genes that make those proteins that we saw or some of them anyway, so quite a few of them come from the nucleus, but quite a few of them also, well, 13 of them, encode those proteins that we saw. And when those proteins have got mutations in them, here you get dysfunction of the mitochondria. So the mitochondria aren't able to generate the energy that they were previously able to do. And all of this is research that's only come about in the last few years. And probably in the last 6 or 7 years has been this term mitochondrial heteroplasmy. And this is what we're going to hear more and more of in the future. And I just want to explain what exactly mitochondrial heteroplasmy is. So if you can think of normal mitochondria, and let's hope we've all got healthy mitochondria within our cells, and we can represent them like this. Unhealthy mitochondria with those mutations in the DNA are like this. And so a normal cell here is called a homoplasmic cell. And it was thought actually that healthy people would have homoplasmic cells. Actually, the more and more we look into it, it seems that we've all got some degree of heteroplasmy within us, some degree of unhealthy mitochondria in us. And so what happens is, if you've got just a few of these unhealthy mitochondria with these mutations inside the cell, you get mild symptoms and various diseases. And if you've got a lot of them, you get severe symptoms and diseases such as Parkinson's, etc. And these are called heteroplasmic cells. And what's really interesting is that it's now being shown again and again and again that this mitochondrial heteroplasmy lies at underneath many diseases. Recently reported it's involved with age-related macular degeneration, which is the number one reason for loss of vision in developed countries in later life. Parkinson's disease, I've mentioned, Alzheimer's has been implicated there, multiple sclerosis has been implicated there, osteoarthritis. But really what I want to emphasize is this is a relatively new research area. It's only just blossoming because it's only now that the techniques for actually analyzing mitochondrial DNA exist. So I do believe that many more areas are going to be found or diseases are going to be found which involve mitochondrial heteroplasmy. And also, I think it could be that mitochondrial heteroplasmy also underlies much of aging as well. So I was introduced to this by a chap called Dr. Daniel Ives, who came out of the University of Cambridge, and he was working on mitochondria. And he said to me, there's this thing called mitochondrial heteroplasmy. I said, I've never heard of it. And then he told me what I've just told you, and I became fascinated by it, and I started reading about it, and I thought, you know what, I think Daniel's onto something here. And he said, I want to form a company. So I said, okay, I'll help you out. So we started Shift Bioscience. So we've only just started it. It's only been going just over sort of 6 months, and we've only capitalized it with a tiny amount of money, but it's still really, really exciting. And the basis of Shift Bioscience is this, that in all of our cells, we've got a mixture of healthy mitochondria and unhealthy mitochondria. And what if you could shift the ratio to healthy mitochondria using drugs? So the whole point and purpose is that so as we age, we get more of these unhealthy mitochondria that are producing lots of free radicals and little energy, and they're a bit hopeless. What if we could shift those back to the healthy mitochondria and select out these ones so that these multiply in an evolutionary process? And this has been shown, so Daniel's shown this routinely, and it's not just Daniel, it's well published. You can actually see this. It's all well published now. But this does actually work. So it works when you use human cells in cell culture. It's never been tried before in any other organisms, but we're just about to go into mice with some of our early stage compounds. So that's all very interesting. So I'd like to just summarize there then. So I hope that I haven't depressed you, because we are living longer and healthier lives. And this aging research is enormously exciting and really will help alleviate some of these diseases of aging. So we're going to have healthy We're going to have health spans increasing and lifespans increasing. There are really promising compounds in clinical trials, despite the lack of funding, but nowhere near enough money is being spent on aging research. The FDA really does need to recognize aging as a disease. And finally, mitochondria, I hope I've convinced you that this term mitochondrial heteroplasmy is one you're going to be hearing more and more of in the future as its star is rising. So I'm going to finish there. So thank you very much for listening.
Well, Jonathan, that was absolutely riveting. Thank you. I'm going to shut up and ask for questions. We'll go on for 10 minutes because we started a little bit late and I'm sure there'll be plenty. So if you try and catch my eye, I will try and fairly sort out the order of batting. And there's a gentleman by the rail at the back there, please. And if you could give your name, if you'd be so kind, before you speak.
Sure. Hi, I'm Kareem Ahmed. So I was very curious to ask you about your point that not enough money is spent on aging research. Do you think that's kind of entirely dependent, basically, on the FDA's decision, or are there other things that can be done to increase the resources, for example, by governments or something like that?
No, it's actually not entirely dependent on the FDA not recognizing it as a disease. One of the problems we've had is that this way of thinking about aging and how those diseases which will eventually kill most of us are researched. So it's just it's almost like a paradigm shift of thinking. Well, if we can delay aging, we can actually stop a lot of these diseases from occurring. And that paradigm shift, and that was led by. First one to really do that was Aubrey de Grey. So my hat off to him. He was the one, and everybody made fun of him. But now it's becoming mainstream that actually this is a really viable approach.
Gentleman here, please.
Hi, William Baines. I've got observation and a question. Observation is that Aubrey now looks a lot older than he does in that photograph.
Sorry?
Aubrey now looks a lot older than he does in that photograph there. So I don't think he's taking the drugs. But I think you're right. Regardless of the FDA recognition, pharma companies aren't going to develop this because it won't be reimbursed. Because if it was reimbursed, then everybody on the planet is aging and everybody would try to get drugs for it, and the insurance system would break, particularly in the US. So given, and it's probably broken already. So given that, who is going to pay for this? And how? But yeah, who?
Well, if you look at the compounds that I put up, I mean, Metformin has been off patent for years and years, so it's as cheap as chips. NMN, nicotinamide mononucleotide, at the moment is very expensive because it's only manufactured by one manufacturer, but as more and more people use that, it will become cheaper and cheaper, and again, it will be really, really cheap. So you could actually imagine that if these clinical trials do show a positive effect, you could have governments advising much the way they do with statins, saying, let's deliver these supplements and drugs to as many of the population as we possibly can. And it will cost next to nothing. These are very, very cheap compounds to produce. So that's one way of thinking about it. But that's the government route. And then pharmaceutical industries will probably not like that very much because their whole business model is treating diseases of aging. They get a lot of money from that. So there might be a bit of a backlash there, if I'm honest.
Hello, my name's Simon Menther. Sort of following on in the same area, do you have any comments to make about low-dosage aspirin? As I believe it's been...
I think that's a jolly good idea. Yeah, I think that's been well researched, and actually, in fact, that is a really good example of something that's very sensible, and GPs prescribe it Of course, it's got a few problems with internal bleeding, and it's not for everybody. But it's a personal choice. But it's certainly the science is pretty robust with it.
Dan, 2 rows down on your left.
Hi, I'm Jim. Thank you for that. In one of your earlier slides, you had the sort of mortality and eras graph. It seemed there's sort of a 15-year window in every era where we go from sort of 60% of people dying out. Bearing in mind the mechanisms you're talking about, do we know why it seems to occur in that 15-year window? What activates all of these age-related diseases? And why that's moving right along your graph over time? And what's going on with that bottom 5% of people who seem to be surviving way past the rest of us?
That's a good question. Well, I don't think, I do know, actually, and I don't think many people really do know what's happening there, if I'm honest with you. But that extrapolation going forward was one where it actually is what could happen if all of this aging research comes to pass and we do start treating people with these compounds that can delay aging. But as to the reason for the 15-year window, I don't know. Sorry.
I was thinking about the reduced calorie intake, and it seems over time we'd be having more calories, and yet we're still moving right, which, I don't know, I was trying to work out how if there's anything you've seen that explains that.
That's a good question. I don't know. Yeah, because we're eating much more, and yet we're living longer. Yeah. Yeah.
Thank you. Thanks very much. Rod Kirkby. As a type 2 diabetic, I'm familiar with metformin. And an interesting thing is that if you take an antioxidant with metformin, it doesn't work. It actually relies on increasing the reactive oxygen species. The other thing I was going to ask, though, as an engineer, when I first discovered telomeres, I thought, oh, that's brilliant. If I was an engineer and I wanted to put a time span on something, I would do it with telomeres that shorten each time. And then you reach the end. You haven't mentioned that at all, so just interesting to see what you think about the telomere thing.
Well, actually, the telomeres were like really hot science sort of 20, 30 years ago. And they still are, but they've kind of fallen out of fashion once these pathways that I mentioned, the SERT pathway and the mTOR pathway, are sort of leading the way. But there are companies out there that are engineering telomeres. There's one I can't remember the name of, but their CEO engineered all her telomeres. And we'll see how she gets on. So yeah, telomere research is very, very interesting, but it's kind of been dwarfed now by these other pathways.
Thank you.
Hi, I'm Arkham. Thank you so much for your talk. So you mentioned that because it's not recognized as a proper disease, you don't get funding for clinical trials. But have there been efforts to use the same drugs for patients with progeria, for example? Because that is the genetic disorder, HGPS. Because if that is recognized as a proper disorder, maybe there'll be more funding to use the same drugs to see how aging progresses in patients with progeria.
For which disease?
Progeria, so the one where you age faster.
Yeah, I honestly don't know.
Let me find the full name, sorry.
Okay. I know that there's work on MELAS disease, but I don't know about progeria. Yeah.
So, let me see. It's called the Hutchinson-Gilford progeria syndrome. It's a genetic disorder, basically, where you age faster.
I honestly don't know.
It's okay.
Yeah, sorry.
Thank you.
Sorry.
Hi, my name is Kumar, and I was curious what your thoughts were around Google's Calico company and the fact that they're actually getting a lot of criticism from the scientific community, being that they're very secretive, and but on the other hand, they do have a lot of the financial resources to sort of make breakthroughs. Just wanted to hear your thoughts on Calico and what you thought.
Yeah, I mean, Calico's, there's a number of these companies that are springing up. I don't know an awful lot about Calico, if I'm honest with you, but because they're secretive. Yeah, good luck to them, but I don't know if they're going to succeed or not.
Yes, hello. Thank you for your talk. My name's David Thompson. I'm just interested to know, sort of left field, is there any similar research being done on psychological components of aging? I'm just thinking that I remember the time when my father, at about 91, 92, decided he was old, and that was it. And also, because he lived to 92 and 93, I've kind of decided, that I can live it up, because I've got it in my genes, but I probably will die early, just because I thought that. I just wonder, are there connected psychological components to the aging thing? I mean, I think there are, but I'm just wondering, in your experience, is there any research into that?
I don't know of any research, but anecdotally, I was sort of thinking that today, because I was thinking of Ronnie Wood from the Rolling Stones. So he, has been diagnosed, he's just done drugs all his life, diagnosed with cancer, etc. But now he's a recent father. He's got two young daughters, I think, that were sort of very young. And he seems to have got a new lease of life, despite the fact, he's probably aged himself prematurely. He's 70 years old. And through his hedonistic lifestyle, he's certainly damaged himself because he got lung cancer. But he seems to be fitter than ever because suddenly he's been rejuvenated. So whether that's the psychology kicking in, I don't know, but yeah, I think it's got to be something to do with it. Because it used to be thought, again, that the immune system was sort of detached from our brains. And actually, there's loads of research now that actually, our immune systems are intimately dependent on how we feel. And how we feel, if we're healthy psychologically, we have healthier immune systems. So, on that count, it's got to be right, hasn't it? So, you're not aware of any kind of funding or research? I'm not, no.
Well, look, that's a very happy note to end on. Partly we can think ourselves well, and if we throw in a bucket of mitochondria, we can live forever. I shouldn't be flippant. Jonathan, that was an absolutely stonking talk. You could see from where you were standing that the room was absolutely riveted. I can't imagine anything with greater clarity, greater interest, and for those of us of a certain age, some immediate interest. Thank you very much indeed.
Thank you.